Hello, my name is Barbara Harrington. Today's date is March 27th, 2018. This week's lab, we're doing the biochemical testing for microbial identification. This is part one of our lab. After today, we're going to incubate our broth for 48 to 72 hours. So that's why I'm doing it today. With part one, culturing the MRVP broth. All right, so before I started this lab, to prevent any contamination, I did 10% bleach and wiped down my whole area. I have my alcohol, my isopropyl alcohol, and my undiluted bleach ready for my lab. The first thing we're gonna do is this week we're doing the E. coli and our S. epidermis, which we have been culturing since week one. So, with our E. coli sample, we're going to take our E. coli, and then I already labeled my MRVP nutrient broth, the MRVP 5 milliliter broth. I'm going to open up our E. coli broth that is already been incubated since week one. I'm sterilizing it, so I just go over the flame. I'll put the cap down right here. Then I'm opening up my MRVP of the E. coli broth. This has not been inoculated yet. And I'm also gonna sterilize that. I'm gonna make sure I also don't cross-contaminate the lids. So first I'm gonna take my pipette out of the alcohol, kind of run it through, let it air dry, squeeze it, use a paper towel, give it about 30 seconds to air dry. This is just help also with the sterilization process so I don't cross-contaminate. Okay. Now I'm going to pipette without touching, sorry, there's a little bit more alcohol, without touching the sides of my nutrient broth of my E. coli. I'm gonna go down and try to get all that stuff at the bottom, the sediments at the bottom, so I have a good amount of my broth. All right. Then I'm going to just drop 4 drops from my cultured 1, 2, 3, 4 into the MRVP broth. Can use the rest of it and put it back into my nutrient broth so I can save it for later. For my next week's, I'm gonna put my pipette that I used into the undiluted bleach. Then I'm going to sterilize both of my vials before screwing on the cap. And I even put the date on it so I know that I started incubating it at 3:27, and I'll check on it at 3:30 for part 2. There's my nutrient broth. Okay. Now I'm going to do the same thing for my S. epidermis. So again, I want to sterilize, running it through the flame. Okay. My inoculated, this has not been cultured yet, of my MRVP S. epidermis broth. Okay. Sterilize over the flame. Make sure I don't cross-contaminate the lids. Now I have a new pipette that was in alcohol. Just gonna, again, dry it off. Get all the alcohol sediment off. Okay. Use a paper towel to dry the rest off because I just don't want to contaminate my specimen with alcohol at the same time. I just want to have it sterile. Again, I'm going to go down to the bottom to try to get the sediments up. Just need a little bit because I'm only doing 4 drops. And then 4 drops into my MRVP broth. 2, 3, 4. All right. I can put the rest back because I'll need those for the following weeks. Put that in the undiluted bleach. Then again, I'm going to sterilize before capping them, making sure I don't cross-contaminate the caps. Sterilize. And then I also wrote the date on this one. 3/27 is when we inoculated it. And again, we're gonna check on it in 48 hours, see if there's any growth in our MRVP, and then allow another 24 hours for a total of 72 hours to see if we have any growth of the incubation time. Thanks for tuning in.